Hello students, this is your biology teacher Pradnya Nigde. Today we are going to deal with the development of monocot and dicot embryo as well as their comparative account. So let's start. Let's begin with the definition of embryogenesis. The process of development of zygote into an embryo is called embryogenesis. We have already seen the embryo is developed at the micropylar end of embryo sac. The growth of the endosperm, it only triggers the development of embryo. After fertilization, the embryonic development begins. Now, how does this take place? We will see in the next slide. Now, the zygote, it forms a wall around it and it is converted into the oospore. The oospore divides and it forms a two-celled proembryo. The larger cell towards the micropyle is called basal cell or suspensor initial cell, and the smaller cell towards the chalaza is called terminal cell or apical cell or embryonal initial cell. The suspensor cell divides transversely, means horizontally, in one plane, and it produces filamentous suspensor of 6 to 10 cells. The first cell of the suspensor towards the micropylar end, it becomes swollen and it functions as the hostorium. The lowermost cell of suspensor is known as hypophysis, which is shown by the small letter h in the diagram. The suspensor helps in pushing the embryo in the endosperm to ensure the nutrition of the embryo. The embryonal initial cell, it undergoes 3 successive mitotic division and it forms octant. Now, how come after first mitotic division, embryonal initial cell, it produces 2 cells. After second mitosis, it produces 4 cells, and after third mitosis, it produces 8 cells. So thus total 8 cells are formed. The planes of divisions, they are at right angle to each other. So these 8 cells, they are arranged in 2 tiers. The lower tier of 4 cells of the octant gives rise to hypocotyle and radical, while the upper 4 cells of tier, they form the plumule and cotyledon. The number of cotyledons, it varies in monocot and dicot embryo. Number of cotyledon in monocot, you know it is 1, and in dicot it is 2. The hypophysis by further division gives rise to the part of the radicle and the root cap. Later, the cells in the upper tier of the octant divide in the several planes and they become the heart-shaped. After that, they form the lateral cotyledons and the apical or the terminal plumule. The further enlargement of the hypocotyle and cotyledons result in a curvature of embryo and then it appears horseshoe-shaped, shown in the diagram G. Let's see the various parts of the dicot embryo in capsula. This diagram, it is there in your textbook, so you can refer the same. The various letters, they represent the various parts of the embryo. We will see them. Capital A, it represents the oospore. Two-celled proembryo, it is shown by the capital letter B. Small e, it represents the embryonal initial cell, as well as small e1 and e2 are the cells which are formed after mitotic divisions of the embryonal initial cell. Small m represents the embryo sac membrane. B1, it represents 4-celled proembryo. Diagram C, it represents the further development of the embryo. Capital S stands for suspensor. Small h, it stands for hypophysis, which is the lowermost cell of suspensor. Then Capital E, it represents the embryonal mass, which is in italic print. Then diagram D, it shows the LS of ovule. Now the various acronyms used. Endo represents endosperm, anti for antipodal tissue, embryo, it shows the developing embryo. Then E diagram and F diagram, they show the various stages of embryo. F diagram represents the LS of ovule. After that, G diagram, it shows the horseshoe shaped embryo, and H diagram, it shows the mature seed. Now, once again, the acronyms. COT, it means cotyledons, hypo, hypocotyle, rad, radical, RC, root cap. After that, H diagram, which shows the mature seed, in which PL stands for plumule. In H diagram, endosperm has been totally consumed, so it is not shown. After that, so what we have seen, diagram E represents heart-shaped embryo, diagram G represents horseshoe-shaped embryo, and H represents mature seed. This is a diagrammatic representation of various stages of development of embryo. The embryo development, it is similar in both dicot and monocots up to the octant stage. The difference appears later. In monocot embryo, single cotyledon occupies the terminal position because it is a monocotyledonous plant. Mono means single. The single shield-shaped cotyledon you can see in the diagram, it is known as scutellum. The protective sheath of plumule, it is called as coleoptile. And the protective sheath of the radicle is known as coleorhiza. Finally, ovule is transformed into the seed and ovary into the fruit. Now, let's compare the monocot and dicot embryo. Monocot embryo as well as dicot embryo, they both are a rudimentary stage of their respective plant. Now let's see the difference between them. Monocot embryo, it consists of only one cotyledon as the name suggests. Dicot embryo, it consists of two cotyledons. Second point, a single monocot cotyledon appears at the terminal position in the monocot embryo, whereas two cotyledons occur laterally in the dicot embryo. After that, plumule occurs laterally in the monocot embryo. In dicot embryo, plumule occurs distally. Next point of differentiation, sheath of the plumule, it is known as the coleoptile in monocot embryo, whereas in dicot embryo, plumule has no sheath, so there is no coleoptile in the dicot embryo. Sheath of the radicle, it is known as coleorhiza in monocot embryo. In dicot embryo, no coleorhiza is present. Now, the next point of differentiation. Cotyledons in monocot embryo, it resembles the true leaves, whereas in dicot embryo, cotyledons do not resemble the true leaves. Now, let's see what is the future of each part in the embryo into the seed. Let's start with the integuments. The integuments of the fertilized ovule persist and they get transformed into the seed coat of mature seed. The outer tough and resistant seed coat is called as the testa, and inner thin membranous seed coat is called as the tegmen. After that, what is about the nucellus? The nucellus in the ovule may persist in some genera like black pepper and beet as a thin papery layer called as the perisperm. Next to that is the cotyledon. Cotyledons in some non-endospermic seeds act as a food storage, and in others they are the first photosynthetic organs. Next to that, micropyle. Micropyle persists as a small pore in seed coat to allow the entry of water and oxygen during the soaking. This is how the ovary begins to differentiate into the fruit and ovary wall develops into the pericarp. Thank you. If you are having any queries, please feel free to ask it me.